going to be looking at meiosis 2. We've already looked at meiosis 1, so we're just going to pick up where we left off in our previous video. And so meiosis 2, we're going to focus in on is this second stage running along here. We have already done meiosis 1, so if you haven't watched that video, it's really important that you watch that first, and then you can return to this video now. That has also a recap in it of what exactly meiosis is and how it's similar to mitosis. So looking at prophase 2, which is the next stage in our meiosis journey, is you will notice now that we're dealing with two cells at the same time, remembering that we started with one cell, we took its chromosomes, we exchanged the DNA, and now we've divided it into two separate cells. We now need to take each of these two separate cells and we need to separate the chromatids from one another, because then we'd have too many pieces of information or too many pieces of DNA in the cell, and so we still need to do that. Now, prophase 2 is a very short phase. It doesn't have a lot of detail in it. There's no crossing over taking place now, and that is because the chromosomes are no longer homologous partners. You'll notice in the diagram alongside that the homologous partners have actually been separated from each other, and we know that based off the picture because we find one of our homologous partners in this cell, which is this purple chromosome, and we find the other one in the other cell. And because they're in separate cells now, they can't actually cross over with each other anymore. And so now what we're going to do now is in prophase, we're going to look at what is its purpose and how to identify prophase 2. So the overall purpose of prophase 2 is to allow for our spindle fibers to form and our centrioles to move to opposite poles. Very importantly, it's to note the following things about what to look out for and how to identify it by what's not happening in prophase 2. So how do we identify prophase 2? We are looking for the fact that, number one, there is no crossing over taking place. And that is because the homologous pairs are now in two different cells, so they can't get to one another. The second thing that we can use to identify prophase 2 is the recombinant chromosomes are visible. And what we mean by that is if we look at the diagram that we have alongside here, you can see that the chromosomes have little bits of another chromosome on them. They've got a little bit of purple on them or a little bit of blue. And now that means that they're recombined. They're not their original form. The last thing that we're going to look out for is obviously our spindle fibers forming, which is what we also mentioned in the purpose, but it's a nice indication to show how far along we are in the phase because the fibers are now forming. Now moving into metaphase 2, our chromosomes are now doing their characteristic movement to the equator of the cell, and so now they're lining up along the equator. The spindle fibers have now attached to our centromeres, and what we're going to do now is we need to pull apart the chromatids. Right now, if you remember, we have one chromatid, centromere in the center, and then we have the other chromatid on the other side. And what we're trying to do is we want to pull apart these two chromatids so that they separate from one another. And the reason why we're doing that is because we want to make sure we keep the number of chromosomes from this point onwards consistent. We don't want to half the amount again. In other words, we don't want to take 46, then into 23, and then half that 23, which would end up our cells having uneven amounts of chromosomes. So what we need to do is we need to ensure the correct lining up at the equator and then separating. So what is the purpose of metaphase 2? So at this stage, we want to look for random arrangement once again, but this time of our recombinant chromosomes, because now they're lining up as individuals. You'll notice in the picture that they're not touching each other like they were in metaphase 2. They were touching their homologous partner. They were still bivalents. But here, they're individuals. And so what we want to do is we now want to arrange them at the equator once more. We're not really sure which side of the chromosome we're going to get. In other words, what I mean by that is, are you going to get this top section or are you going to get this bottom section? That is another form of random arrangement. Now, how do we identify metaphase 2? We identify metaphase 2 by looking for our double-stranded chromosomes aligning at the equator, which that's exactly what they're doing in this picture here. They're lining up in the middle of the cell. And secondly, we're looking for those spindle fibers attaching to our centromere, that structure that holds the chromosome together. And remember, we ultimately want to now pull it apart. Now, this then brings me to anaphase 2, which we're going to look at now. But I want you to be very cautious. A lot of students get anaphase 1 and anaphase 2 confused with each other. Now, starting with anaphase 2, we can already see some dramatic differences. The first thing is you will notice that there is our characteristic space that is appearing in between our now separate chromosomes. And now some terminology gets a little bit tricky because previously we had one chromatid with a centromere in the middle and the other chromatid on the other side. And this whole thing, the whole structure, we called a chromosome. But the moment you pull two chromatids apart, like we're doing here in this diagram, we now refer to each individual strand as a separate chromosome. So technically, in this diagram, I've labeled them. That number 1 is one chromosome, and that is number 2, a separate chromosome. I know that they used to be called chromatids, but the moment you pull the centromere apart, they're now individual chromosomes from one another. So, how and what is the purpose of anaphase 2, and how do we identify it? So looking at the purpose, we want to separate our recombinant chromosomes. Remember, recombinant chromosomes is where now they have these little extra bits of other chromosome attached to them, these little areas of different color. And we want to form single-stranded chromosomes. Chromosomes cannot exist in their duplicated form forever. They have to be single strands at some point. Now, in order to identify anaphase 2, we need to look for spindle fibers that are contracting, in other words, they're getting shorter, and that these double-stranded chromosomes are becoming single. And we can see that, as I mentioned earlier, where I labeled it 1 and 2, those are single-stranded chromosomes. They used to be attached to each other, but now they are separate. And now finally we enter into the final phase, telophase 2. As you can see here, we've had a lot that has changed. The first thing you'll notice is that a nuclear membrane is starting to form around each of our new sets of chromosomes that we have freshly separated from each other. And so essentially what that means is we've taken 2 cells and we've now turned them into 4. Each one of these will produce a separate cell. And so as we can see here, the nuclear membrane is reforming. And all we need to do now is to pinch off the cytoplasm. If you remember what that looks like, it's when the cell does this and we pinch off the cytoplasm via cytokinesis, and we will make 4 separate cells. So the purpose of telophase is really straightforward. It is simply to go through cytokinesis, where we pinch off the cytoplasm via the cell cleavage. And then how do we identify telophase 2? We're going to look for 4 separate cells that are non-identical. And you can see that these cells are not identical in front of you here because of the colorations that they've used. And last but not least, we are going to look for single-stranded chromosomes. In other words, we are going to look for structures that are one single chromosome. We don't want to see 2 chromatids attached with the centromere. So in other words, this is not what we're looking for. We are looking for individual pieces. Now my diagram here still has the cytoplasm attached to one another, but essentially once the cytoplasm has pinched off, you should have 4 cells in the picture as well. Sometimes they give you one like my diagram here, sometimes they'll give you the 4 individual cells. So let's make a quick summary of meiosis 1 and meiosis 2 collectively. At the beginning of meiosis 1, we started off with a cell that was sitting in interphase. You'll see that we have 2 single-stranded chromosomes. They went through DNA replication and they doubled the amount of genetic information inside of them. Remember, we do this because we have to double the genetic information at first so that we can half it later. We also go through interphase to double the genetic information because we want to make sure that each of the cells gets the correct chromosome number. Remember, we want to go from a cell that has a full set of chromosomes that is diploid, and we're trying to make a haploid cell. We now move into meiosis 1. And here we see our chromosomes moving towards each other. They are going to go through the process of crossing over, random arrangement, and at the end of meiosis 1, we've done two very important things. The first thing you'll notice is that our chromosomes now look a little bit different, and that's because of crossing over. The second thing you'll notice is that we started with 2 chromosomes originally in the cell. We now have 1 each. And so at the end of meiosis, you will produce 2 haploid cells. Now, we can't leave our cells with these chromosomes in them while the chromosomes are still replicated. In other words, you'll see that these cells here still have their centromeres, and we need to get rid of those. We need to essentially take the chromatids apart. We need to move them away. So now we enter meiosis 2. The purpose of meiosis 2 is to take these chromosomes, which we call recombinant chromosomes, and we want to separate the individual strands, the chromatids, from one another. And so what we do is we take each one and we place it in individual cells. And you will notice that each of these 4 cells contain 1 chromosome. So it's important to remember that we originally started off with 2 chromosomes. We finish meiosis 1, and now each cell has 1 chromosome in it. Now, even though we separated the chromatids from each other into separate cells, which we don't call chromatids anymore, we call them single-stranded chromosomes, at the end of meiosis 2, every cell still only has 1 chromosome in it. Often that's the thing that gets most confused about, because when is it a chromosome? When is it a chromatid? Remember, if there's a centromere, it's still a chromatid. No centromere, we now call it a chromosome. And so, at the end of meiosis 2, we are going to have 4 unidentical cells, and they are haploid from the original diploid cells that we started with. So to round off our lesson, let's do our terminology recap. So random arrangement is when our chromosomes line up at the equator and we are going to separate them. In this instance, in meiosis 2, we're not separating them from the homologous partner. Instead, we are separating them from their chromatid partner. 1 chromosome, therefore, goes into each cell. Cytokinesis is the separation of our cytoplasm, and at the end of meiosis 2, you should have 4 separate cells. Diploid is when you have a full set of chromosomes that have not been changed, whereas haploid means it's a cell that has half the amount of the original chromosomes. An example of this is the diploid number of chromosomes in humans is 46. The haploid number is 23. Recombinant chromosomes are chromosomes that have gone through crossing over, and now they need to be separated from one another in meiosis 2, because we need to separate the individual arms of the chromosomes, which in all instances we can also call them recombinant chromatids. And last but not least, single-stranded chromosomes. That is the goal of meiosis 2. You want to take a double-stranded chromosome that has been recombined, and you want to transform it into a single strand. If you don't do this, your cell will have too much information inside of it, and you can end up having an abnormal meiosis result, which is not what you want. Thank you for watching, everybody. I hope this was very helpful and informative video. This one was a little bit shorter than the previous one. If you liked it, please let me know, and I'll be back very soon again to do more explanations. Make sure you go through the other videos, look at perhaps the exam questions that go with these topics so that you can prepare for tests, and I will see you again. Bye.
Yeah, give me a slice and you know it. Now I'm in slowly. I put a smile every day in your face, yeah. I'm putting everything in order as you say, yeah. I was in love with the crazy, yeah. Just get inside of my way, then we can do it. Taylor is never in a rush. Shout out to Facts for the trust, but to the mommies, bend the trees, but to me, play the beat.